Okay, let's continue the exercise for this part. In your question book on page 4 and 5, there are human ones, there's one for plants, one for bacteria, and the last one is on assembly. Spend about 10-15 minutes on those. Yeah, that's right. Okay, we had about 20 minutes here. Does anybody want more time to finish one exercise? I think we can leave it there. I do not expect you to do all of them, and I'm happy if you can do one at a time. Is there anything I need to clarify? Is there anything I need to show on the exercises? Shall I demo one? No? You're happy with the exercises and using the answer book for those?
Where to find the CRISPR track?
Where to find the CRISPR track? You type in CRISPR into the search box and you find the CRISPR track.
How do you know if the CRISPR are allowed any kind of digestion? I don't know how to realize that.
Okay, yeah. I may go for that and show that. So, CRISPR exercise. Doing a quick demo of exercise 2. So this is the CRISPR sites. I select human and go to this region. And then I add my track, so I go to configure this page and search for this track. Which is called CRISPR. I put it on with labels. And you can see, I make this a bit smaller. You can see you actually have 2 tracks. One at the top here. These are the CRISPR sites on the forward strand. And there's another one here, which are the CRISPR sites on the reverse strand. And for this exercise, you really have to only look at whether these sites overlap the phenotype causing variants. And the phenotype associated variants are in this track here. So you've got this blue one and then these ones. So you essentially just line up. So this pink block here is a variant which has a name. If you click on it, you get the name. It's a deletion. And then you can say, okay, this deletion overlaps with this side here, and then this side and this side. You do the same for the other ones. Yeah? Cool. Any more questions on exercises? No. I will reset my configuration now. And then we move on to looking at genes and transcripts now, where we have a presentation first. Okay, so in this part, I'm going to introduce the different gene views in Ensembl. I will explain our annotation methods, then give some indications of how you can assess the quality of a transcript, and I will then also introduce our system of stable identifiers, and finally gene ontology, which we use to classify gene functions. If you've done the exercise 1, you will have looked at BRCA2 gene already. This is how it looks in detail. Most of the genes we have several transcripts, and transcripts is just a term that we use. It's the same as isoform or splice variant. So now these combinations of blocks and lines are the different BRCA2. The protein coding transcripts are shown in red, the non-coding ones are shown in blue, and then we have so-called merged transcripts, which are shown in yellow. And these are the high-quality ones, which I will explain in a moment. In the transcripts, you have exons and introns. The exons are the boxes. If they are colored in, they are coding. If they are not colored in, they are non-coding. Or they can also have untranslated regions, which are not colored in either. And the introns are the connecting lines. From this view, you can also tell that BRCA2 is encoded on the forward strand, and you can tell this by the fact that it's shown above the blue line with the context. Also, those arrows next to the labels that point to the right. If they pointed to the left, it would be a reverse strand. And likewise, the introns point upwards. If the introns point downwards, that again means reverse strand. We use two different annotation methods to annotate our assemblies. On the one hand, we have the automatic pipeline, which is run by the Ensembl team for all the species that we have in Ensembl. And on the other hand, we have a manual annotation, which we do for human, mouse, rat, and zebrafish, and that's done by the Havana team, which is part of Ensembl by now. So for the automatic annotation, we run pipelines on a genome-wide scale, and these algorithms use experimental data from a variety of biological databases to make these predictions. This is the evidence that we use to make these predictions. We use nucleotide data from the INSDC, which is the International Nucleotide Sequence Database Collaboration. And that is a collaboration between ENA at EBI, GenBank at NCBI, and the DNA database of Japan. They exchange data on a daily basis. In addition to this data, we also use protein data, which comes from UniProt. And finally, we use the manually annotated proteins and mRNAs from RefSeq. These are the ones that start with NP or NM, and we only have these for human, mouse, and zebrafish. For a variety of other species, and also for the 16 mouse strains we have, we, in addition, infer genes based on homology with other species. As an example, the majority of the genes in the chimpanzee genomes was annotated by mapping from the human genome. And increasingly, especially for new genomes, we use a lot of RNA-seq data for annotation as well. The manual annotation done by the Havana team is also done on a genome-wide scale. This involves a group of people who look at sequence data, reads papers, and uses a variety of sources from public databases to annotate the genomes. They have just finished the human genome, so they have finish the full annotation of the mouse genome recently after the human genome was already done. So these are the two genomes for which the manual annotation has been completed once. They also do this for pig, for rat, and for zebrafish. This manual annotation has advantages, so it tends to be more comprehensive. You get more genes per genome and more transcripts per gene, and it's in particular good for regions which are very difficult to annotate with automatic pipelines, namely untranslated regions and highly variable parts of the genome. Manual annotation has disadvantages as well. It's a lot slower. It takes on average half a day to annotate one gene, and therefore it can only be done on a small scale. Otherwise it would be too expensive. We have for 3 species also included imported annotation from sources which are, well, closely linked to us because we know them very well. But yeah, I don't think you work on these. Now, with regards to the quality of the transcripts, I was mentioning the merged transcripts earlier. So this is the example for BRCA2, which has one of these golden transcripts. And these are transcripts where both annotation methods agree, and therefore we consider them to be of higher confidence and quality. Related to this, there is a gene set which is used by many large projects, and this is called GenCode. That's the gene set that we have for human and mouse, and it's a merged set of the automatically and the manually annotated genes. If you want to work with a sequence set where most people around the world agree on the annotation, you may want to use this one here, which is called consensus coding DNA sequence set. And this means that for these transcripts, the EBI, the Wellcome Trust Sanger Institute, UCSC, and NCBI all came up with the same annotation. And you can show these in green in our browser. We have we, EBI, have been continuing this collaboration with NCBI, and recently released the first version of a transcript set where we agree, so NCBI and Ensembl EBI agree, and we defined one clinically relevant transcript per gene. And this set is called main select, version 0.5, and we only have this for 53% of the genes yet, and it's only available for human, but this is the transcript and the transcript set you want to use if you only want to use one. Where this is not available, or for other species, you have more options where you can look at different flags, as we call them, to decide which transcript to use. And you can find these indications for which transcript you should use in the transcript table, as shown at the top of here. So this is an example for a gene called Bambi, which has, see on the right hand side, we have got flags here. The first one I explained, main select, so if that is there, you definitely want to use this first transcript here. If that's not there, you go from top to bottom to the things on this slide and decide based on these indications. So in addition to main select, we have the Priest principal isoforms. This group defines the major isoforms based on protein structures and alignments. This is available for a variety of species, and they score these isoforms from 1 to 5, where 1 is the best quality. You can also look for a full-length protein coding transcripts with the GenCode basic set, another flag, and then furthermore, for human and mouse, zebrafish and rat, you look for these two columns here, so the consensus coding DNA sequence and the biotype, which gives you the colors I have explained, so yellow or golden, meaning it's a merged transcripts where the annotation methods agree. And then finally, if you work with a species that doesn't have any of this, you would need to go for the transcript support levels, which range from 1 to 5, and where 1 is the best quality. That's again a flag in the table. So all of this should help you decide which of the many transcripts of your gene to use. It's not always as easy as in this example. Sometimes it's a bit ambiguous. And then you will need to go into the supporting evidence for all of the transcripts and look at raw data and sources and how much data there is for any given transcript. And I will do that in the demo. Okay. Good. To keep up with all the data that comes in constantly, we work with a system of stable IDs, which are exactly what they say. They are stable, so they don't change over time. They take a certain format. They start with an ENS and then a G for gene, T for transcript, and so on, followed by a number. If you work with a non-human species, you have an additional 3 letters here, which represent your species. These stable IDs are important for you in the same way as the release number, because you can always go back to them and find your gene or transcript of interest again. The problem with gene names, for example, is they can change over time. These don't change. So please write down your stable ID for any kind of thing you work with in Ensembl, and it will help you to find it again. Yeah, and there's a publication on this gene annotation system here. Questions on gene annotation? No questions? Okay. Before I go to the demo, I will start for the first time today to introduce an ontology. And we use a system called gene ontology, or in short GO, to describe gene functions. Why is that necessary? Well, if you talk to people about their research or gene of interest, somebody may say, I work on innate immunity. The next person says, I work on non-specific immunity. The next person says, I work on cytokines, and so on. There are 2 problems here. One is that people use multiple terms to describe the same function, and the other one is that the gene descriptions, they tend to be very specific. So this is where a gene ontology comes in, to help you to ensure that everybody understands you and you talk about the same thing. These Go terms, in short, form a controlled vocabulary of terms. So here's an example where we have the term innate immune response, which is associated with an accession number, shown here in bold, and then you can look up this term under this accession number, and you get a very specific definition of what that term actually means. In addition, these Go terms are also hierarchical. This example, innate immune response, is a daughter term of immune response, and it itself is a parent term of all these other terms related to innate immune response. So what you can actually do is you go into this hierarchy on a website called geneontology.org, and you can see where your term fits into this hierarchy as well. We would very much encourage you to include these Go accession numbers in your papers if you write about genes. It helps other researchers as well as database annotators to link functions to genes. Questions about ontologies? No? Okay, so that was the first example of an ontology. We will have more later. We're going to have another demo now, where we will start looking at a gene, which is ESPN. And this demo starts on page 19 of your course book. Okay, I'll go back to the homepage. I select human as my species and type in ESPN, the gene name, and click go. My search results are ordered by how commonly used the pages are. So our top hit here is human gene. We have restricted already to only human. We can restrict to different categories as well. So we could say we are only interested in genes, transcripts, and the like. So now I select ESPN, human gene, my top hit. And this opens the gene tab, which is the second tab we are visiting today. And on the left-hand side, all the menu items are now related to this gene tab. On the top, you have got your symbol and your stable ID. For human, this tends to be HGNC. And then we have the location of our gene, which is on the forward strand, and some more information about that gene. I will visit the sequence view in the menu on the left first. This shows me my sequence in a FASTA format. So I've got my sequence header, which includes the assembly, followed by a colon, and then the chromosome, which is 1, followed by a colon, start position, colon, end position, colon, and then another 1. And the 1 here at the end is the strand. So this is the gene on the forward strand. If it's reverse strand, it would be -1. And then we have our sequence. The sequence is color-coded, so we highlight all exons with a yellow background, and then the exons of the genes we're looking at, here ESPN, with red letters. So that's an ESPN exon here, and this is a neighboring exon that overlaps. You can configure this page to show less or more of flanking regions. For this, you go to configure this page, where you can change these options. You could say you don't want to see flanking sequence. You could say you don't want to see variance, which is what was loading on my page there. Yeah, and other options. There's a download button at the top that enables you to either download the sequence in FASTA format, which I've just described, or in a rich text format, which you can open in a word processor such as Microsoft Word, and this includes all the colored highlights. You can also blast this whole sequence using the blast button, or you could highlight a part and drop that into blast. Okay, so now I'm going to have a look at the gene functions, and for this I will visit this ontology section here, which is separated into 3 parts. We have Go biological process, which shows you what this is on a protein basis, so this shows you what the protein does. And then you have Go cellular component, which is where it is located, and then Go molecular function, which is how it does it. For each of these, you get a table such as this with the accession number, and then the Go term, the transcript it relates to. And any of these tables, in fact, any table in Ensembl, you can filter by using the filter button at the top. So you can filter for something in this table, and likewise you can download it and open it in Excel using that button there. So now we know our gene is somehow related to locomotion. We may want to look at in which tissues it is expressed. For this, you go to the menu item gene expression towards the bottom. And this brings up a little display that comes from Expression Atlas, which is also at EBI. So you see this heat map here on the right hand side, which at the top has the different tissues, on the left has the different experiment types, and then these colors here relate to how strong the expression is. You get the numbers if you mouse over them. What you can also see if you mouse over them is that in the body map on the left hand side, the tissues light up in this body, and you can do that the other way around as well. You can select the tissue here, and it will be highlighted in the heat map. This is a male body now. You can change it to a female body, and you can also have a similar interface for brain tissues if you're interested in brain tissues. Likewise, you may want to filter this table by your organ or anatomical system of interest using the button at the top. You can sort it by expression ranks, and you can of course download it as well. Okay, so this was one example of external data that we pull in. If you want to find out about this gene in all external databases, you can go to external references in the menu on the left, further down, and this shows you a table with all the links to other databases where this gene is mentioned as well. Expression Atlas being one of them, but you also have HGNC, MIM, which we will have a look at later, NCBI, and more. And these are hyperlinks, so you can go to those original resources then. So now I'm going to leave the gene tab and go to a transcript. For this, I will click on Show Transcript Table at the top, which shows the whole table for this gene. You can read at the top, this gene has 18 transcripts. So if we had to decide from this table to use only one of them, I would choose the second one from the top because it has CCDS, UniProt, GenProt Basic, and APRIS, but also it has a transcript support level of 1. So what I will do is I will click on the stable ID for this transcript, which ends 28.5. And that opens the, I think I went back there. And that opens the transcript tab, our third tab, with our menu now being transcript specific. Again, we have got the name and the stable ID at the top, the location of the transcript, and some more information about it, including that it contains 13 exons. I will now find out a bit more about how this transcript was annotated by looking at the data that we used to annotate it. And this is found under supporting evidence towards the bottom of the menu. And here you get a graphical view of your transcript at the top in this golden color with the exons and introns. And then each of these other tracks here relates to one type of data, one data item that was used for the annotation. And all of this is color coded, so you've got the proteins in yellow, the cDNA in green, and the ESTs in purple. You can click on the labels of these tracks to get more information and then to view the original record in the external database. And likewise, you can also click on individual colored blocks here. So I will click on this one here, which is a clone. Sorry, I got distracted there. Which is a protein that comes from UniProt, and I can go now and look at the UniProt record by clicking on this ID here. And I'm taken to UniProt protein database to get information about the protein. You can see here that UniProt has also annotated with a score of 5 out of 5, so it's a high confidence protein. I will close this again and go back to Ensembl. And I will now have a look at the sequence views on the transcript tab. We have 3 different ones, one for exons, one for cDNA, and one for exons, and leave the others for you to explore. So I click on exons. And here I get a table with my 13 exons plus upstream and downstream sequence. And in this table, I've got my exon number in the first column, then my exon ID, starting with ENSE, start position, end position, length, and the sequence as such. Don't worry about these colored variants here, so the individual colored nucleotides. We come back to those later. For the moment, you can look at the exons and introns related colors at the top. So we've got in the table, we've got flanking sequence in green, just here. And then we have a UTR in orange. We have a translated sequence starting with the ATG in blue. And then we go into an intron, more translated sequence. And on the other hand, we also have a UTR and a flanking sequence. As for the sequence view on the gene tab, you can customize this. So you can say, I want to see full introns, for example, or only exons, which I won't show now. And once you've done that, you can, of course, download this as well. We also have links to external references on the transcript tab. They come in two different options. You have your general identifiers to other databases, and which is similar as on the gene tab. And you also have external references to microarray probes. We map a variety of probes to our transcripts from different commercially available microarrays, Affymetrix, Illumina, and some others. And on this page here, you can see now from, for example, this array here, this probe here was mapped to this transcript, and it also maps to some other transcripts, which you can explore here if you like. So far, most of what I've shown is nucleotide data. We also have some protein data, and we are expanding on that right now. You can find this under protein information on the left. There's a graphical interface under protein summary, where you can see your protein stable ID at the top. And then in this picture here, the top row, the top track, is our Ensembl protein with the exons highlighted in light and dark purple, and all these other tracks here represent protein domains from different other databases such as PFAM. So this black domain here coming from PFAM, for example, is mapped to our first 4 exons here, and the same for the other protein domains. The same information is available in table format as well under domains and features. And finally, for some but not all proteins, we also have 3D protein models. For this one, there is none, but we will have one in the exercise. So this is like an interactive 3D model you can turn around and so on. Yeah, so that's the end of this demo. Questions on genes and transcripts? No? Okay. Shaking your heads, I think that means no. So, then we can continue with exercises, which are in your question book on page 6 to 9. There's a whole range of exercises for different species. So you've got human, bacteria, plant, fungi, mouse, and an extra one looking at interactive pathway displays as well, if you like to have a look at that. So yeah, please pick the species you're most interested in and attempt one or two exercises. I included a lot so that you can pick what you like. Okay, I need see here. Yeah. Okay. Okay. Thank you so much.
How many years?
Yes. So this is more in the sense that How do you get on? Do you need more time to finish one exercise? Does anybody want more time to finish one exercise? No? Okay. Then I will ask if there are any questions. Do I need to show anything? No? Okay. We will then move on to variation now. I will start with the presentation again. So I will introduce our system, how we classify variants and their consequences. I will talk you through the species for which we have variation data and where that data comes from. We will then browse that data in different hubs, and after lunch also use the variant effect predictor, or in short, VEP. In Ensembl, we differentiate two broad types of variants. We have small scale variants which affect just one or a few nucleotides. These are most commonly single nucleotide polymorphisms or SNPs, but they can also be shorter insertions or deletions. On the other hand, we have structural variants, which are large scale rearrangements in the chromosomal structure, often copy number variations, but also deletions, duplications of parts of chromosomes, insertions from one into the other, and translocations where parts are swapped. All of these variants, and we again use an ontology to classify these, which is called sequence ontology. To start with variants and coding regions, these can either be synonymous or non-synonymous. The non-synonymous variants are the ones that change the amino acids, so these are called missense variants. The synonymous ones don't change the amino acids. Variants can also affect the UTRs of exons on both sides. They can affect splice sites, introns, upstream and downstream regions of a gene, including regulatory regions such as promoters. This is a table from our documentation that summarizes all the different consequence terms with term, the description of the term, and then the link out into the sequence ontology with the accession numbers, where you can again look at the hierarchy of these terms. So you can see there are quite a few. To make this a bit easier in the browser, we color code our variants. We also run 6 different algorithms to predict the effects of the variants on amino acid sequences. So these are called pathogenicity predictions. We only have them for missense variants because these are the ones that change the amino acid sequences. It's important to note that all of these scores provide only predictions, not facts. And if you see anything interesting there, you need to follow it up experimentally. This is an example here of a variant table where I've got variant IDs here, some information about the variants, their consequence terms, so they are missense, and then these are the columns relating to these pathogenicity predictors. And you can see we use a traffic light color system there, where green means the variant has a benign effect on the protein. Yellow means it has a very red means it has a very severe effect, and yellow somewhere in between. What you can also see is that these scores not always agree. That is expected because they use different algorithms. and we recommend that you just use this as an overview for a set of variants. With a bit more detail, these are the first 3 scores that we use, SIFT, Polyfen, and CAT. They use a scale from 0 to 1 or 0 to 100, and different methods which are outlined at the bottom. SIFT, for example, uses sequence homologies and chemical similarities of amino acids. So for SIFT, if you have a change from a leucine to an isoleucine, it would have a score near 1, which means tolerated, because they are quite similar biochemically. If you look at the scales, I think it's clear that the people who developed these algorithms didn't speak to each other while doing so, because these scales are sometimes opposite. So the benign scores for polyphen are near 0, while the tolerated ones for SIFT are near 1. We do SIFT scores for all of these species. All the others are just available for human. And these are the other 3 scores that we use. Revel and MetaLR both use a variety of tools and then combine the results into one score. This slide summarizes the species for which we have variation data in Ensembl. This, of course, includes human mouse, zebrafish, Drosophila, yeast, but also many farm animals and primates. We also have variation data for 26 species in Ensembl genomes, including some plants such as beans and tomatoes. We don't have any variation data for bacteria, though, I'm afraid to say. We import that variation data from a variety of different sources, and we quality control it, we link it to other data, and we display it in the browser. And this slide summarizes the sources of our data. The major source for short genetic variants is dbSNP. dbSNP is actually a misnomer. It not only includes SNPs but also other short variants. And then we have dgVA for the structural variants. We have European Variation Archive and NextGen for animal data. And moving on further, there are some databases which focus on certain diseases or phenotypes. GWAS catalog, for example, or COSMIC, which is the catalog of somatic mutations in cancer. We import from those as well. And finally, we have variants which are on certain SNP errors from a couple of companies too. With all of this variation data, in particular if you use it in clinical settings, it's important to know what the baseline is in healthy people. And that's what the 1000 Genomes Project has done. They looked at variation in 2,500 individuals around the world. This project has finished some years ago, and they were looking at people from different what are called superpopulations. So these relate to different continents: America, Africa, Europeans, East and South Asians. And what you can do then in Ensembl is to go into this data and have a look at the allele frequencies for any given variant in any of these populations at a continent level, but also then at individual populations within the continent, and all the individuals as well. And that enables you even to go then and follow this up in the lab, because from these people there are also cell lines commercially available. Our major source of allele, it's the genome aggregation database, or GNOMAD. From this, we have imported data for more than 140,000 individuals. These are, again, from different populations, which are summarized on the right here. Some people were genome sequenced, others were exome sequenced, some were healthy, some were diseased, so it is really a mix. What I want to point out on this slide, though, is the graph here on the left. This summarizes the number of individuals in this and other projects, such as EXARC, that's the precursor, and the ancestry of the people. And you can see that people of European ancestry are clearly overrepresented in the studies, and that's still the case, and it is a problem for research and diagnostics, and there are ongoing efforts to get this right, to produce this kind of data from people from non-European populations, including specific populations such as Native Americans. You will hopefully remember from this morning how our reference genome is not a genome of a single person. This has some importance for how we think about reference alleles. So if you go back to this example here, where this lady BL contributed samples where then the DNA was used to sequence and generate this contig here. If we assume that she had a T at this position in this contig, we will call the T the reference allele. And if there is a G in other people, we would call the G the alternate allele. And we write down our alleles as reference / alternate, or in this case T / G. So our definition of the reference genome is just based on the very fact that she was the person whose DNA was used and she had a T. It has nothing to do with the frequency of the allele, so in this case the T is a minor allele. It has nothing to do with whether or not that allele is the ancestral allele present in other primates, and it also has nothing to do with whether or not it causes the disease susceptibility. Here, in this example, it causes the minor allele, the T causes the disease susceptibility. Okay, so what I'm saying here, in short, in summary, is reference allele does not mean it's the major allele, and it does not mean that it's not associated with a disease either. Okay, are you happy with this so far? Good. Then I will add to this the allele strand. So variants can not only affect genes transcripts on the forward strand, but also on the reverse strand. And this is important for how we then define them in our database. In this example, we had a TG, but we're looking at a transcript on the reverse strand. So this means actually that in the exon there's an AC. In Ensembl, we always write down our alleles as if they are on the forward strand. So we would have in our database and in the browser a TG, while in this case of a reverse strand, in the gene it's actually an AC. DBSNP does this similarly. They either would say it's an AC on the negative strand or it's a TG on the positive strand, which is fine, it's unambiguous as well. They are moving on over to only reporting on forward strand 2. The problem starts when you read the literature. In cases like this, you will find either AC or TG without information about the strand of the gene. And that's then a situation where you go into a genome browser and check the strand of the gene, and if you can't find out, you may need to ask the authors what they're talking about. Okay? So yeah, my message here is if there's anything about the confusing about alleles, check for strength of the gene. Okay? Good. Any questions here? Yes.
You mentioned that the variation of data comes from, for example, in the human gene phenotype, and you know that there are two kinds of databases. One is which is for free, and the other one
Yes.
do you have both data? No.
We only use what is freely available. Yeah.
Yeah.
That's a good question, actually. Yeah.
You mentioned the difference between the reference allele and frequency.
Yeah.
Is there any chance to modify these, the reference allele, to coincide with the most frequent allele?
Yes, it's actually an ongoing process. So when we move from GRCH 37 to GRCH 38, we started with that. So the reference allele, which were minor alleles or only alleles in the reference sequence in one single person, were replaced by the most frequent one. And we continue to do that for, it's ongoing.
By something that you're doing it on Ensembl?
Oh no, not we. We, as in the Genome Reference Consortium, is doing that.
Okay.
So they are doing the assembly and the reference sequence.
Okay.
They are replacing the minor alleles with the major ones.
So it's about that when we established the 38 assembly to, for example, annotation variants,
Yeah.
we should get a decreased number of variants that would. I'm thinking about the lot of variants that we see using the 37.
No, el número de variantes sería el mismo. Lo que tendría es que cada tener. No, la variante siempre se ve la misma. Lo que va a cambiar es que el número de variantes mayor del 50%.
That part I didn't understand. That was too
Sorry,
fast.
the big change is the number of variants above 50%.
Actually, I don't think no, because if it's your reference, if the reference changes and your individual has the reference, which wasn't reference up to
You mean your sample against the reference?
exactly. When you perform the annotation, you're going to lose most of these variants. Frequent.
Yeah,
Okay,
quite
yes.
possible.
Yes.
Yes, you map
Yes.
your map the variance against GRCH 37 and then it's 38. Yeah, you may find less variance with them. Yeah. And some, well, talking about those two assemblies, sometimes you have variants reported on 37, which are not reported on 38 in the literature, and the other way around. And also the location changes. So it's problematic if you move over, but we help you. We have tools for that. We have a conversion between the two if you need to. More questions? But if you start fresh, use 38. That's the assumption. More questions? No? Shall we break for lunch? Yeah? Okay. Let's do lunch break and come back in an hour at 1.
No, cortamos este cal.
¿Cuál otro va?
Doce.
12. 12 conectados.
Yeah. Both going to.
Volvemos en una hora.
Yes.
Volvemos en una hora.
I will leave the cable something here.